So, a summary of where we are at. So I showed you in the previous lecture something about single bacteria, and then we decided to look a little bit more into what happens if bacteria get crowded around surfaces. And I introduced yesterday this idea that these colonies of bacteria are quite interesting. They differentiate, they look like multicellular organisms, but we don't have a good understanding of why they're doing that in the sense of what are the functions connected to gene expression. Okay, and then we had this idea that one of the main things that biofilms do is they excrete polymers, and this production of polymers might help in spreading. And then I asked you whether you would think that as a consequence of this, of the water flow from the substrate to the biofilm would swell or expand, and everybody said expand, but then it wasn't clear what the forces were. And then we had this great proposition by your colleague that maybe gradients, horizontal gradients in polymer concentration could create gradients in something. And we are trying to find out whether that is true and quantify this to see the shape of the biofilm. Okay? And then, well, to make this, to do some math about it, we had to do some assumptions. And so I'll walk you through what we did yesterday. Is we assumed that the biofilm was a material. So we put under the rag a lot of the things that Lisa told us, and we looked at this as a macroscopic material, okay, where things would change because the bacteria would produce polymers, and this would change their concentration in space and time, and hence encourage water to move one way or another by changing its chemical potential. Okay, chemical potential and osmotic pressure go hand by hand, and this is the way you see it. So assuming that this biofilm is made of two fluids, the biomass and the water, you can write down a complex functional, which is called radiant, or which is the dissipation plus the free energy change. And if this is a slow departure from equilibrium, you can get the equations of motion by extremizing this functional with respect to the velocities of biomass and water. This is all we did yesterday. Then we calculated those derivatives, and we got to an interesting point where at least we understood the physics, which was this combination of four equations that were quite transparent. One is the Stokes equation for the biomass. So I remind you, the Stokes equation is the limit of the Navier-Stokes equation, or Reynolds zero. Okay? This, I didn't remind you about this yesterday, but again, these non-dimensional numbers keep coming back. When Reynolds is zero, you can neglect inertia, and then what you get is a linear equation for the velocity in this limit, and this is called the Stokes equation. Okay, this is all stuff we've done yesterday, so you don't have to, it's just to remind you where we are. And then water would move through this network of polymers and cells through a well-known law, which is called Darcy's law, and it's typically used for understanding how water flows in porous materials, right, where you have pores of some kind created by the structure of the system, and water flows through it. Okay, like water permeating through the soil would be described by this equation. Okay, so this is quite nice because now we understand this is the engine for our entire dynamics. So cells produce biomass. This deviates the system from equilibrium. It causes gradients in osmotic pressure, and this is what puts the biomass in and the water in motion. Okay, so we understand that the engine is the production of biomass, and we can calculate everything else starting from there. So that I told you that there was a black box, which is just a black box because I don't have time to do all of the calculations. But essentially, by assuming a bunch of things, you can reduce the system of four equations into just one PDE for H. H is our object of interest because it's the shape of the biofilm. So if we solve for H, then we'll know the answer. We'll know whether the biofilm grows vertically or horizontally, and depending on what parameters. Okay? So this was a black box, but at least qualitatively, from this equation, we know that this produces both behaviors, both vertical growth in the absence of this term. This is given by this exponential growth, okay, and flow, because this, if you have done any thin film model, you recognize that the form of this is a flux, okay? So if you define the flux as everything that's inside the parenthesis, this is how the shape of the biofilm will vary as a function of biomass growth rate and flux. Okay, so if the biomass flows, then its shape, the shape of the biofilm, overall shape of the biofilm will change. And this you can recognize it from thin film models of like a fluid going down a slide driven by gravity or whatever else. Okay? And then our goal was try to find the solution for that. And we were almost done. So I'll remind you where we are. Until here, things make sense. And then I found out yesterday that if I write down here, people in the back don't see. So I'm really mad at you because I asked you. Mad, really mad. A bunch of things happened down here and you missed out. But anyways, okay, where we are at is, okay. So we start from this equation. Yeah, sorry, there's no way I remember to I hope so, because there's just no way. It comes, maybe at least I'll do HT, so it's at least consistent, but this will keep coming. So a reminder, DTH equal HT, DRH equal HR. H is a function of both R and T, and it's the height of the biofilm as a function of distance from the center. Okay, so this is R, for example, and this is H of R at this time T. Okay? Okay, and so we did a bunch of tricks. First of all, we would eliminate this dependence, this term, but assuming that H can be written in this way. And then we normalized implicitly time by assuming that there would be another time variable, that reabsorbs this term. Okay, and so by the way, I realize I didn't say it explicitly, but this means that you know what dt tau over dt is, right? This is why it's implicit, because I don't know what r of t is, but in principle, if I had an equation for r, then I could plug this back in and have an equation that's closed. Okay, so this is the derivative of the new implicit time variable with respect to the original one. Okay. And so then I won't redo all of the steps, but we found already the solution for R, which was great because it already told us a lot of information. And I'll write it down in this way. Okay.
Yeah, so, I mean, it's not often one sees a square root of 7. No.
Well, in this kind of problems, it's not so, like if you have a drop that expands, I think like the singularity in time is a t to the 10.
Okay.
So is there a way to understand the 7 I would have to think about it. I mean, one bottom line, and I keep getting these questions, is I'm pretty sure that I could make scaling arguments, and I've not done that. But since you've seen the system, so it's not obvious how to make them work, so I should definitely do it, but this will be for next time. Okay, this already told us an interesting thing, which was that this variable r, which we haven't really defined well, it's a kind of extension of the biofilm, and we have assumed that my solution would rescale with respect to distance from the center if I divide it by this variable. So it's not a very well defined, it's just a radius of the biofilm. Okay, this would have two regimes, right? Initially, it would remember its original radius, and then expansion would kick in the moment where growth was large enough. Okay, so at a certain timing point, this would be much larger than the initial condition, and then you would start expanding. Yes?
Maybe it's minor, but isn't that term like 1 over the arcane expansion?
D tau equal dt dt over d tau. Correct.
Well done.
Okay. Thank you. What else did I want to say? Okay, and so the last part of the equation, so I remind you, the moment you make the assumption that this H is self-similar, and this is the correct ansatz in this geometry, then if things work, then your equation will decouple in something that will only depend on tau, which was our equation for r, and something that will only depend on our non-dimensional variable psi. We called it psi. It's r over big R. Okay, and we got to being almost done. I realized I didn't separate the variables for you, but it was already separated. So maybe I should get back to the last line of your notes from yesterday and redo it. So we were at this point, I think. Okay, so we were at this point, and well, this is already separated actually. I don't remember what point we were at, but I got this question. I wrote it down there. Maybe we were at the previous. Does this make sense with your notes from yesterday?
Yeah, I think we had that. You should maybe just rewrite psi. Yeah. I have this equation.
Exactly this? Okay.
Well, no, but like the R10s were in, but
Yeah, we're here.
inside the position of the psi's and the R10s, but yeah.
Okay. So I'll assume that we got here, and this is separated, right? You can assume now you can bring this on the other side if you want, and this only depends on psi, this only depends on tau, so the only way this is true, this equation is true, is if this is separately a constant and this is separately a constant. Okay? Separation of variables, just classic. Okay, so now we've solved this already, and now we'll solve that one. And we'll solve it by recognizing that this is actually a derivative. Okay? And surely I'm missing a prime somewhere. I'm definitely missing a prime here.
No, in the other one.
Correct, in the other one. Sorry.
And you're missing a prime, I think, on the other side, outside of the parentheses. Oh, but you have the
Yeah. Okay.
Yes.
Other convention.
Third notation.
Sorry, but this is because this is actually interesting the moment you realize f only depends on psi. So this looks like a partial derivative, but in fact it's not, right? Okay, so this is the reason why this sometimes comes up. Do you want to vote? Do you want me to try and use prime or psi?
No, but then you could use a prime instead of that.
I know. So I think this is the right one.
Yeah.
People
Yes.
did.
Yeah.
Is this better?
Yeah.
Okay, perfect. Sounds great. Okay, so now this you can very easily solve it because you recognize that this is a derivative. This is the derivative of what? There is a minus that actually, can I do this without you killing me?
No.
You're dead so many times over.
Yeah, exactly. I'm dead already, so that's fine. Actually, no. Let me let me leave it like that because otherwise it will be different from my notes. And since okay. Okay, so this is a derivative, right? Great. So now this equation is very easily solved. We can do, does this go up?
Yes.
Yes. Okay, great. So now we are solving this equation. Plus a constant. Okay.
Minus sign.
Others? I think this is good. And now we have to impose boundary conditions. And when psi tends to 0, that means r is going to infinity. And that means that this f needs to go to 0, right? Because this is conserving mass. So as your psi goes to 0, means you're flat, completely flat, and your f goes to 0. So this is equal 0 by boundary condition. Okay, so now we keep going, and now there's 2 of these, and 1 of these that go away. This goes away. Okay, and so we have f prime equal, there is a minus, and then there is a psi. Okay, and again, there is an f square here. What else am I missing? I think this is right. Okay, and this constant will be fixed by the mass of the biofilm, right? That's the other boundary condition, so we'll just call it C. Okay, we won't compute it, doesn't matter. Fixed by mass conservation. Okay, and so we have this form, self-similar solution is F equal 3C, but we'll call it C because it doesn't matter, as I said. Let's say that this is C prime, so you know that there is a 3 in between them. And then 3/2, and then let's write psi explicitly. It's R over R square. Okay?
Where R itself is a function of t.
Yes. Let's do T because we've solved it already. So this plus this completely solves the problem. Okay, that's the dependence on time, and this is the dependence on space. Okay, now a couple of comments about this shape. So first of all, this is obtained by making assumptions. and particularly the self-similarity. Self-similarity typically holds if you are far enough away from the initial condition, but still you should verify, right? All of these arguments tend to be self-consistent, which means they're not really proofs in the mathematical sense, but they make sense. And then if you want to be sure, then you do numerical simulations, and if both agree, then you're okay. Okay, so first of all, you simulate that equation and you get this solution and you're happy. Second, if you look at this shape, now if you had to draw it, this...
Can I ask, because I mean we did prove, you know, there was this moment of happiness yesterday when we proved that it is self-similar. So we made the ansatz and then we...
Well, we didn't prove that it is self-similar. We proved that if you make this ansatz, so if you assume that it's self-similar, then the solutions, the equation separates, so you can solve it.
Okay.
So this is still an assumption. It's a
we self-consistently, I mean that we don't go outside of the space of solutions of the ansatz, right?
yes. So it's self-consistent in the sense that it doesn't prove your ansatz. An ansatz is always an ansatz.
Yeah.
Typically, if you solve it, then you know it's nice because it will be true in some regimes, right? But typically, if you start with a very broad biofilm, it takes some time to forget about the initial condition. So typically, this kind of self-similar solutions will be true at long times. And you might want to check what is that long time. Typically, it would be the time for the solution to expand more than the initial radius. And that's true in this case as well, because...
But that, I mean, to do that, you find the time for which the self-similarity breaks down, right?
Yeah,
How would you find the time below which...
from the R0, so you would take that and say, what is the R such that R divided by R0 is much larger than 1? This is for sure, I mean, it's not for sure, but this is what typically is the regime where this works. And all of these things are not exact, right? So it's important that in actually, sorry, just 1 second, in all of the asymptotic arguments that you make in your life, then please remember, make arguments, be bold, be bolder actually. So you have to really, you make these ansatz, you have no idea whether it works or not, but if you find a solution, that's already good, because then you can start thinking whether this is, you expect this to be right or wrong in some regimes or others, but the bottom line is then you couple asymptotic arguments with numerical simulations, and that's bulletproof. Not mathematically, but we're not mathematicians. Yes?
Let's see, how can you find out that f is equal to c minus 2 over 2?
How you get this?
No, I'm talking about the parameter c.
The parameter. So you have to integrate your solution. So now we know you can start, let's start from the equation that doesn't have g. So that's more clear because that's mass conserving. So that's the equation for h. And you'll do the integral of h for r that goes from 0 to infinity. And this is in cylindrical coordinates, so you also have to multiply by this. This will be a constant. This you have to input into your problem, right? It's a boundary condition. If you start with a biomass that has, with a biofilm that has this mass, then you can compute C, because you plug your solution into the integral. We know that we've done things correctly, so it will be constant, and it will only depend on C. Okay, so it's only a matter of doing the integral with this 1/3, and it will be, okay? Okay, so first comment was made. The second comment is if we were to draw this solution, do you see any problem? R is never, I mean, small r is it doesn't matter because it's at the numerator. Small r equals 0, that means that you started with a delta and, you know, sure, this is singular. But yeah, but let's start with a finite r, okay? So yeah, that could be a problem, but that means that you yourself put yourself in a situation where the biofilm was quite weird. So yes, that could be a problem. Let's not put ourselves in weird situations. There's something very simple about this equation that might actually be wrong. Correct, it's to the 1/3, so this will go negative. So this requires you to match the self-similar solution. So let me write down the self-similar solution in the original variables. So we have it. Okay, so there is an a e to the gt, okay, and then big H will be f, which is this guy, c minus 3/2 r over r squared to the 1/3, and then divided by r squared, and I'll just write r squared. Well, let's not write it down. We have it upstairs. C. Okay. So the bottom line is this equation, this solution goes negative, so you have to match it to some boundary conditions. Okay. And maybe at this point I should show you some data to motivate what boundary condition I choose initially. Yes, this is the self-similar solution we just calculated, so then you have it on the slides if you need to go back, and this should be right. Okay, so actually maybe at this point let's talk about how to compare the theory with the experiments, since I'm showing it to you. So we just said that our solution predicts that there is a transition, if k is small, from initial swelling to spreading. Okay? And this is what you see in the experiments. So what I'm doing here is I'm marking, so these are experimental profiles of a biofilm. They're done by looking at how much light is transmitted through a biofilm, and then matching the light, having a calibration between transmitted light and height. Okay? This is the center of the biofilm, that's the edge, and these are different times. And you see that initially the biofilm just swells. Okay? And then eventually it starts spreading. Okay? So the experiments show the transition that we have predicted. I remind you, just starting from first principles, writing down the idea that gradients could create forces, we found out that there should be this transition. Okay? So this is great. Also, the profiles will tell you what is the right boundary condition, at least at these stages. So you see, if you go after the bulk, this is the bulk of the biofilm, and then there is this layer of cells. This layer of cells, at these locations in the biofilm, the position that we got yesterday of cell-cell contact, that is where this applies. Okay, remember I asked you what forces could cause the biofilm to spread, and somebody proposed that just by growing, cells would push against each other, and that would create a force. This is where that happens. Okay, this is almost like it starts with a monolayer, and it remains pretty packed until you get to the bulk of the biofilm. Okay, so this has a different dynamics than the one that we just described. And as you can see, this layer that is in front of the bulk initially grows vertically, okay, because it has this different dynamics. So the boundary condition I chose here was the following. Let's say that this this self-similar solution applies until the point where H reaches H infinity, and H infinity, sorry, H8, H infinity, okay, it's this height, where H infinity has a different dynamics, and it's growing, in fact. H infinity at time 0 times e to the gt. Okay, this is just motivated by experimental observation. If you do this, now you have a better grasp at what R is, and this is something also that is typical in this kind of calculations. You have a better grasp of what R is because you can compute what is the maximum slope of the biofilm. Okay, remember, if we don't do any matching, the maximum slope goes to infinity. Okay, if we do this matching, we don't get the slope going to infinity. We have a finite slope that connects the biofilm to the boundary condition. Okay, so this point is quite well defined, both theoretically and experimentally, and it is the point that you see marked here. This is the point of maximum slope. Before you define this, it is very hard to compare theory and experiments because you don't exactly know what R is. And you know there are these two different dynamics. You know that yours applies to the bulk, but how do you define the bulk, especially when it starts expanding, is not really obvious. Okay, so this is a way to make this clean. Okay, and then so you can compute the slope and then the predictions. So you have everything in here. I'm not going to do more calculations because I think this is enough, but the prediction for the radius is that it will start from a constant and then go up, okay? And I'm writing the prediction as a function of gt normalized with the growth rate, and the prediction for the slope is actually very neat because it does this. Okay, as a function of time. So this is the moment where you start spreading. This is swelling and this is spreading. And you understand it. You see it from there and you understand it. As you swell, the biofilm goes up, but the radius is not moving, and so the slope will increase. And then as you start spreading, the slope can decrease or increase as a function of, it can decrease to 0 or remain constant as a function of what this H infinity is doing. So you see that at long times, you have to worry about this H infinity. Right now we were quite sloppy, but then eventually, and you see it very nicely from here, eventually the dynamics at the contact line will really matter. Because you see that initially, when the biofilm swells vertically, this layer is doing its own thing. It doesn't matter for the bulk. But the moment the bulk starts spreading, it reaches the contact line very fast. And so the contact line is what determines how quickly the biofilm will move. Yes?
Sorry, I got confused. In the slope diagram, what's the dashed line representing?
This is going to be, so this is what the model predicts right now with this boundary condition, okay? But I just showed you, and I was telling you before, that this will matter for long times. Because at some point the bulk reaches the contact line, and you cannot neglect that anymore. And so this is an alternative possibility. Okay, so if instead of having H infinity grow, you have H infinity constant, you see a different behavior. And in particular, it does this.
So I understand that the slope goes down when it spreads, but when it swells for me, I guess it swells like this. This is swelling.
Swelling means that you move, H is increasing and R is not changing. So that means that
So the slope
you're higher, yeah. Okay, so this point is defined very nicely from this graph. It corresponds to the point where the biofilm starts spreading, so everything makes sense. So first thing So I don't know how much time I should spend on this, but at least I want to show you what the consequences are for translocation. So, okay, so first, sanity check. This is a mutant that does not produce the polymers. Okay, as you can see, the contact line will move because the cells will grow, so they will push each other, but there's no transition. Okay, so there is no osmotic spreading if you don't produce polymers. Okay, and so here, I'm gonna go fast, but just to give you an idea, yes?
Just to clarify, so at long times at R equals 0, H tends to a constant, like in the experiment.
When, sorry, say it again?
In
When
the experiment, H tends to a constant at R equals 0.
oh yes, sorry, this is saturation, right? Because the biofilm won't keep growing infinitely. And this, of course, occurs because there is a limitation in nutrient.
So like, how do you see that from?
You don't, because I've assumed here you should make a better model for G, right? G is the growth rate, and right now I'm just saying this is a number, but yeah. And there are, actually I should say, there are much more detailed models, especially, so there are much more detailed models that account for height saturation, account for wetting, account for surface phenomena. So you see the picture. This is the minimal model that accounts for our intuition that polymers would help spreading. But then if you want to be quantitative and say exactly, after this transition, how this works, you have to look into the details. H infinity will change in time. This is the initial time. At the initial time you have a monolayer, and then they grow. Okay? Okay, so match theory and experiments. Okay, G is not a constant. We just had this remark, but let's fit an exponential anyways and get a number. Okay, that number, it might be trivial, but in fact, until I did this, nothing worked. The reason is the constant you get here, of course it's sloppy, but the number you get here is 2.3 hours. Okay, so in a biofilm, which is well known, the cells don't grow at the maximum rate that you see them grow in a shaking condition, right? And it's a factor of 4 or 5, right? Bacillus subtilis in a shaking condition grows at a rate of, with a time scale of half an hour. Okay, so you have to get an order of magnitude. And that's the one-dimensional parameter in this theory, right? We have K, which we haven't talked about, but now I'll tell you what it is, and G, which is dimensional. Okay, then we have our prediction for the slope. And as I said, I showed it there. This is the theory which neglects the dynamics of the contact line entirely, and these are the experiments. Okay, the red line, the red points are the experiments. The green line are the experiments with no polymers that don't show the transition. So this curve, so it has essentially two parameters that are my two parameters. One is the width, and that is given by G. Okay, if you have a G that's half an hour, this just doesn't fit. It does like this. It's tiny. Okay? So this is the first one that made me very happy. The second one is K. And what K determines is what is the moment for this transition. So K small will shift that transition, put that transition off. And K large will put this transition at an earlier time. And I'll tell you just in a second why this is true. Okay, now we don't have any more free parameters, and this is how the prediction for the radius and the data for the radius, this is the comparison. And it doesn't have free parameters. This is the APS mutant, the one that doesn't produce polymers. It doesn't work. We're happy. Okay, this is what happened. Okay, and this is just a, yes?
To clarify, the mutant, that corresponds to setting some parameter in your model?
Not really. It's because it's not really, because it just really doesn't have that motor. It could potentially, and because here there are two things, there is the contact line and there is the bulk, the osmotic spreading. It's just in the wild type, osmotic spreading is important with respect to the contact line, and in the mutant it's probably the inverse. So it's hard to make one parameter change that will account for this. Okay, this is just another way to say that eventually you will have to care about the contact line. This is another model, which is essentially what I told you about, essentially the same thing, where you add the surface energy. Okay, so in your functional, you have an additional contribution, and you can write down exactly the same equations, where you now take into account the fact that the biofilm is more or less happy to wet the surface. Does this make sense? When you have a droplet that's spreading, this contact angle will tell you how happy the drop is to wet the surface. Okay, whether the relative energies in the interfaces is larger for liquid vapor or volume vapor.
We didn't
Does
have
this?
wetting in
There's no wetting here because there's no contact line. I assumed that this was the case, and it turns out what the important message in here is that ignore the green line, but there's just one line here that shows up. Sorry. Can you see it? Yeah. Yeah, for some reason it didn't show before. So there are two lines, gray and black, and ignore the green. This is, in this new model, accounting for wettability. When wettability is low, what you see is that the radius of the biofilm will initially increase anyways, but then will level up. So the biofilm doesn't spread anymore. It stops. Okay, this is the result of this paper. At high wettability, you see that there is the initial spreading, and then it keeps going. Okay, I don't want you to understand exactly what the scalings are. Just pay attention to the fact that wettability is only important at long times. Right? These two curves are the same initially. In both cases, the biofilm spreads. Just in one case, it stops, and in the other case, it keeps going. Okay, yeah. What is wetting? It's what I was sketching here, and I was assuming that people knew something about it, but probably not. So,
not this generation. It's like liquid.
not this generation. Okay, what is wetting? If you have a drop on a substrate, it can look like this, or it can look like this. Okay? The reason why there are different shapes for a drop on a substrate is because in this case you are increasing the amount, you are decreasing the interface, the amount of interface between solid and vapor, right? There's less of that there, but there's more interface liquid solid. Right? So why should a droplet decide to be in this configuration versus this configuration? It depends on the relative energies of the interfaces. Okay? So if creating interfaces between liquid and solid is very costly, then you want to be in this situation. If you are in the opposite case, then you want to be in this situation. Okay? So contact angles are the experimental measures that will tell you whether a drop likes to wet a surface or not.
Is it different from surface?
It is another thing. It's connected, of course, but it's different in the sense that it requires to understand what's the energy that's needed to create an interface between liquid and solid and vapor. Okay, so the surface tension will only tell you about the liquid.
So what makes water like high and low? Because the substrate will be the same. So
Well, so maybe I went too fast. This is a different paper in a different situation, and in particular they compared two biofilms that have different wetting properties precisely because they have different concentrations of surfactants. Okay? Yes?
What if you have a previously pre-wetted layer?
Yes, pre-wetting layers. So the moment you add surface tension, it turns out that stress at this point, at the contact point, diverges simply because H goes to 0. So there are different ways to regularize, and one is assuming there's a pre-wetting layer. If you do that, then you cannot have the you don't have that description of the contact line motion in this way. You have to do different things. You either assume that the contact line is sliding on top of the substrate, or you assume that there is a pre-wetting layer. There are two independent ways of regularizing this singularity. They are equivalent, but you have to work it out differently. In both cases, there is a parameter anyways. One is the
Is it just a different way to regularize it, or do you get different physical results?
So you have a parameter anyways. In one case, it's the velocity of this contact line, and in the other case, it's the height of the wetting layer. So sometimes it's more appropriate one versus the other because you know something about the substrate. So the interpretation will be different. Typically, the math, it's just different ways of regularizing the singularity. But physically, if you want to interpret those parameters, then you need to think about what the model was.
Are there physical cases, like scenarios, where you would have a very large pre-wetting layer?
There are situations, I think, in which the pre-wetting layer is something that happens and it's not just a mathematical trick. I'm not aware of, I wouldn't know how to give you an example of where this is particularly visible. For the biofilms, it happens. Sometimes you'll see it, and it's probably driven by surfactant. Sometimes you'll see that there is a clear layer of water, of liquid, that expands outward, and it doesn't contain cells. And it's probably driven by, sometimes the cells will do something particular, because I haven't observed it all the time, so I don't know what triggers it, but it's probably driven by this gradients in surface tension.
When you're measuring this angle for wetting, how do you decouple the wetting effect from the surface tension and the friction of the substrate? Seems like all of these things would somehow be important for
Yeah, definitely. You'll have to be careful with that. By the way, this is really not the topic for this, but you have to be careful. Like, if you do this, then you'll have a friction that is imposed by, you'll have a model for the motion of the contact line, and in that model you'll have to include friction in the way that you think is more appropriate. In this case, friction will be accounted for by sliding, by viscosity. You have a layer, velocity 0 here, you impose a velocity here when the thing arrives, and that imposes the friction. So you don't really have a way to decouple. You have to model everything. Okay. Good? Okay, so we have a choice here because I have half an hour. I think I should skip these guys, unfortunately, because it took a long time to get it. But anyways, so I'll give you the bottom line, and then maybe I'll pass to the next topic, because otherwise tomorrow I don't have the time. Yes.
Like the schematic, like makes me think that did you assume the symmetry from both sides from the center? Because the schematic doesn't show symmetry.
That is perfect. So the next step in our treatment is, what if you don't have symmetry? What if you create a substrate that has a broken symmetry? You put something that makes the osmotic pressure in the environment larger here and lower there. So now we have a theory that works pretty well for determining when you spread, what's the moment that you start spreading, and we can just look at it. Okay? So this is our homogeneous theory. Okay, this is the equation, and these are, I plotted for you the solutions, okay, at different k's. Okay, now I'll have to tell you what k is, but this reproduces exactly what I just told you about here, what we got yesterday from our solution of the self-similar equation, and then I'm plotting it for different values of the parameter. So, number one, here k is larger and here k is smaller. We'll talk about k in a second, just bear with me. So that means that if for some reason I expect k to be larger or smaller in some points on the left or on the right, then I know whether spreading is faster or slower. Okay, k large means spreading is fast. On the other hand, I'm normalizing with g. And g is the motor for everything. So of course, g large means you spread faster. Okay? And so you see it from here directly. If g is large, that means that, for example, focus on this blue one. So fix k. If you increase g, that means that this point corresponds to an earlier dimensional time, right? Because gt is 2 or whatever. So if g is larger, that occurs at an earlier dimensional time. So bottom line, k large or g large means faster spreading. That's our expectation. Yes?
Is there a parameter for, say, how porous the surface underneath is?
For how porous? Yes. So what do we expect for these parameters? Okay. Oh, tell me I wrote the expression for k. I didn't. Oh, fuck. Okay. So I'll write it down here, and it's not in the slides, so... Okay. What k is, so it will be complicated, but we'll try to understand it. Then there is mu water, mu b, and then there is h0 to the cubic. Okay. This is what's k. And I'll tell you what these parameters are, and you'll see intuitively that this makes sense with what we have modeled. Okay? So from my black box, remember, we started from our dynamical equations, and I went into the thin film equation. We had parameters, and these are the parameters that were there. Phi infinity and C infinity are biomass concentration, biomass volume fraction, and biomass mesh size. I'll tell you in a second what it is. At equilibrium. That's why they get the infinity. Okay? Mesh size, what is mesh size? It's the typical distance between polymers in this biofilm. Okay? So bottom line, if you have a very concentrated polymer mesh, if you are in that situation, then your phi will be large and your xi will be small. They'll be very close to each other, and their volume fraction will be very large. Good? Okay, and then we have mu b, which is viscosity of the biomass. This is viscosity of water. This is the initial geometry, h0 at the initial time and r at the initial time. Let's call it R0. Okay? So it makes sense that when K is large, so what was K? Remember, this whole thing starts, do I have the equation? I think I wrote it down. Here. No? I did write it down. Yes, up there. So remember, when K is small, then you don't have flux. You only have vertical motion. And then when K is large, then this starts. Okay, this is what's represented here. How do I expect K to be in this situation where there is a gradient? What do you expect? If the osmotic pressure in the environment is large, what do you expect? You expect the biofilm to be very dense or very loose at equilibrium? The biofilm is in contact with the agar. Let's say that we are on this side of the agar. The agar has a very large osmotic pressure on this side. That means that water does or does not want to leave? Wants to leave? At higher osmotic pressure, it wants to leave? It might, so high osmotic pressure means your view of high osmotic pressure is intuition of high osmotic pressure is coming from the situation where you have a semi-permeable membrane and you have a large concentration here. This means you have a large osmotic pressure. And in this situation, you want water to come in. You don't want water to leave. No water leaves. So on this side of this plate, I expect the biofilm to be very dense because water does not want to leave the agar. This makes no sense? Some sense? Okay, pretty good sense. And on the other side, that's the opposite. Osmotic pressure is low, a lot of water will want to leave, the biofilm will be very not dense. So I expect in this case that K will be very large on that side, on the left, and very small on this side, on the right. Correct? Because dense means this is small, and plus there are experimental results that say that this is also small. Okay? So that's the first arrow. I expect K to be larger in that direction. Oh yeah, I did write it down here, but you can't really see it. Sorry. Anyways, it's that. It's actually even correct. Good. Okay, so large K says that the biofilm should go in that direction. And about G, well, you'll have to rely on experimental data showing you that at large osmotic pressure, you downregulate matrix. Okay, and so these results imply that on this side of my setup, there will be more polymers. So the biomass production rate, actually, there won't be more polymers at equilibrium. There will be more production of polymers. Okay, so G will be large. So there are these two different tendencies, and then this was, the results were maybe 6 years old, and then 6 years later, and I'm really sorry that I can tell you this in slightly more detail because it was really painful. Okay, you have this. Okay, those are, so essentially in a few years we were able to create osmotic pressure gradients in an agar gel, and then we looked biofilms on this agar gel that was heterogeneous. And what we found out is that in control gradients, then the center of mass of the biofilm does not shift in any ways. But if you impose osmotic gradients in the environment, then the biofilm translocates. So at this point, you should be happy because then we are back to navigation, kind of, right? We are moving a little bit at least. Okay, and because I told you all of our expectations, let's look in for 1 second at this graph. took a long time. So this x is the shift of the center of mass averaged over 11 colonies that we got. And y is also the shift in the center of mass, but in the direction that's orthogonal to the pressure gradient imposed in the environment. Okay, that's the convention. X is in the direction of the pressure gradient, and y it's orthogonal. So what this says is that the shift in the center of mass is driven by the gradients in osmotic pressure. Great. If you look slightly closer, you see that there is this bump, and then there is this. Okay? So by the way, positive X is low osmotic pressure, and negative X is large osmotic pressure. Okay? So according to our expectation, this should be driven by G. Okay? And then if you look in slightly more detail at all of our colonies, you see that actually this little bump is real. It's not a statistical accident, but it's real. At least I think I counted 8 out of 11 change their mind. They first go toward positive large osmotic pressures, negative X, and then they move the other way. So this would suggest, and this is the conclusion, that biofilms will first be driven by the physics. K is a physical parameter. It's mesh sizes, it's concentrations. Okay, so first physics will push biofilms on one side, and then biology, so G, the biomass production rate, will change the dynamics and make them move the other way. Okay, so this was a very, very short, introduction to this, guys. Here are, I realize I didn't give you references, but this is a very, very, very short list of things that you might want to check out. And if there are no questions, then we can move to the next topic.
So do you have, I mean, is the a bioarchive paper on this yet?
No, we are writing it right now. But it's gonna happen soon. It's happening
We
now.
should check every day.
Well, yeah, maybe not every day, but okay. So this is a very famous quote from Jacques Monod, and he was smart, but he said this, and I want you to challenge it and tell me why he was wrong. For what we are doing, of course. He was definitely right for something else, but what changed? So we are switching gears now. We are back to individuals. Okay, I've told you in the first lecture that individual bacteria are able to measure gradients at small scales, at small piclet, by measuring differences of chemoattractant along their trajectory. Okay, and then we have this quote, right? And he says that essentially elephants should be measuring time differences in odor concentration to find their mates or something. And do you think this is right? Yes.
So, I mean, if you're going, when we're doing for the E. coli, we ran into small number statistic issues, which you're not going to run into for elephants.
Statistically small
so
what?
when we were doing the chemosensing, for example,
Yeah.
then when we actually, in order to peak noise, you had to integrate over large time, or you had to integrate over space. But then for an elephant, because you do not run into small number issues, you're not going to have, like this was something that Schrodinger said in his What is Life paper. He said, yeah, we are big because we should not be affected by fluctuations in our environment that much.
Very nice, except there is a slight problem in what you just said, which is, sure, there are no, you beat the error coming from counting, right? Counting molecules in a small spatial scale, that's the error bar that you make in the counting process is diffusive. but there is a slight problem about this, that there are other fluctuations, right? That are not coming from the counting process, but they are coming from the signal itself, right? Can you tell me the name of one non-dimensional parameter that will change, at least one, between E. coli and elephants? Reynolds number, and also great. Okay. So this is wrong for what we are gonna do. Okay, and we are gonna try and disentangle where it is wrong and how you can fix it and what is really hard, etc., etc. And then yesterday, talking to people, I think I realized that this transition between small and large Reynolds numbers, small and large Peclet numbers, it might be useful to revise something about this. And I think the 20 minutes that remain might be good for that. And then we'll talk about the algorithms tomorrow. Does that make sense? Okay, so to get a sense, what changes at large Reynolds number? Can somebody tell me a name? Okay, great. How much people, like if you had to give me a sense how familiar you are with turbulence, would you say you are extremely familiar? Kind of, okay, kind of, and very not familiar, no idea. Okay. Okay, great, thank you. That's very helpful. Okay, so this goes for tomorrow for sure. And I did this morning, so it must be here somewhere. Okay, so phenomenology. Navier-Stokes equations? Yes, right? You've seen them. You've seen them. You've seen them. Okay. We'll talk about the Navier-Stokes equations and the equations for the passive scalar. Okay? Okay. So a large Reynolds, what happens is, so what we've done until now in all of the lectures is forget about inertia and only look at low Reynolds number. So when I remind you, Reynolds is UL divided by nu. When Reynolds is much larger than 1, you have turbulence, and where this much larger is typically thousands. Okay, and I'm going to be sloppy about this. I'm not going to cover the transition to turbulence. And this is laminar. And this is what we've done. Okay, so what happens to these equations when you cannot neglect this term? The effect of this term is you can look at these equations in very different ways. I'm going to give you an intuitive version first, and then I'll try to formalize slightly to tell you what is the origin of the features that people talk about when they talk about turbulence. Okay, have you heard about anomalous diffusion, anomalous scaling, sorry, and you've heard about intermittency. Okay, and you would know how to relate them kind of to these equations or no? Okay, good, perfect. So first, intuition about what this term does is the following. If I inject energy in the system, what energy does is it is dissipated. Okay, the Navier-Stokes equations are dissipative. They dissipate energy. Okay, but they do it in a very specific way. They do it because of this term, right? And this term, at very large Reynolds, one would expect it's almost irrelevant. But this is not true. Okay, these equations, their peculiarity is that even if Reynolds is very large, dissipation is not zero. Okay, What dissipation does at large Reynolds number is it's pushed to very small scales. Okay? And so if you inject energy in the system, okay, if you force the velocity to be non-zero, so if you inject energy, what it does as a function of scale, okay, you inject it here, say that you have, I don't know, a large pressure gradient so that creates a large-scale velocity circulation. Okay? If this is turbulent enough, if Reynolds is large enough, this large-scale velocity that you introduced energy at this scale, will do something very special. So it will, initially you'll create these large-scale structures, and then they break down in smaller sort of eddies, which break down in even smaller eddies, and even smaller, and even smaller, and then you get to a length scale eta, okay, which is called the Kolmogorov scale. Gorov scale. Well, energy is dissipated, goes out of the system. Okay, so energy injection and energy dissipation. You've seen this before? Okay, most of you. Maybe I've been optimistic. You've seen this? Okay, good. This range, okay, so we'll call this the scale at which you inject energy, we'll call it L, big L, and we'll call it integral scale. Okay, this range between the integral scale and the inertia and the Kolmogorov scale is very special. What happens here, so I gave you here a pictorial view, but what happens here is, you know, when you are on a plane and you do this, right? So it's chaotic, there's a lot of fluctuations, and these are not square root of dt. Okay, these are major fluctuations, okay, and the entire field essentially is about trying to understand these fluctuations. How does it work at different scales? Can I infer things from one scale to another? And how dramatic these fluctuations are. So the first thing you should remember about turbulence is that there are fluctuations, major fluctuations. Okay? So in this process of creating smaller and smaller structures, you create fluctuations.
So, okay, I just energy is injected at large scales. That's sort of, but it could also be injected at other scales.
In that case, then this doesn't apply.
Okay.
Okay, so
This is where it's
in order for this to be a turbulent, a fully developed turbulent field, you have to have the separation of scales between injection and dissipation. If you do not do that, then it's not going to be turbulent. Okay, but we'll see that L divided by eta is a Reynolds number to some power. So if you are at large Reynolds number, that means that you are in this situation.
And so, I mean, this is a description of an actual phenomenon. So basically, if we do have turbulence in a natural system, we always know energy is injected at large scales.
Yeah, at large scales with respect to the dissipation scale. Yeah, you know that there is a separation of scale. This actually, let's do this right now. So to see how much these two scales are separated, you just have to do, so the first thing you do with these equations, since they are complicated, is you do dimensional analysis. And you make some assumptions and you try and see how far they get you. The first assumption is exactly represented here, which is that the effect of this term is to transfer energy at different scales. Okay? Which makes sense because it won't dissipate energy in itself. If you integrate across the whole volume, this will give zero. Okay, so what it does is it transfers energy at different scales. So let's assume that energy, so if I multiply this equation by V, the order of magnitude of this term, so if in thinking about energy, the order of magnitude of this term will be the velocity measured at any of these scales, okay, and we'll call that R. Let me put this smaller so it's clear. Kolmogorov scale. So if I am at R in between eta and L, the order of magnitude of this term will be, so VR to the power 3, 1, 2, and then I'm multiplying by V because I want to make an argument about energy. And then there is a gradient, okay, will be equal to epsilon, and epsilon will be energy dissipation rate okay. This will be constant throughout this range, which is called the inertial range, and this is a number. Assumption. Okay, let's see how far this gets us. Assume this. Epsilon constant. Okay, and then, okay, since this will occur both at large scale until the Kolmogorov scale, I can equal this too at the Kolmogorov scale, right? At the Kolmogorov scale, it's where dissipation coming from this term becomes relevant. I'm saying epsilon transfers energy from large L up until small l, and then it's dissipated. Okay? So at R equal eta, balance V eta cube divided by eta equal nu V eta square divided eta square. Okay, again, argument about energy, I'm multiplying by V. Order of magnitude of this is nu. There is 2 V's at the numerator and 2 length scales at the denominator. Right? Make sense? Okay. Great. So from this, okay, so these are 2 equations. So I can, let me write this down for at eta, okay. V eta cubed divided by eta is also equal to epsilon, right? On the one hand, I assume that that is a constant and that it also holds at eta. And at eta, I have also the additional balance between these 2 terms. So these are 2 equations, correct? And so we can get eta. And the result is eta. Now I won't ask you if you see if I write there because you lie. Liars. Okay, let's say this is 1 and this is 2. If you put these 2 together, gives eta. Okay. So now we have eta, and we can calculate what's the ratio between L and eta. Okay, because I'm keeping telling you that there is separation of scales. It's important, otherwise you don't get turbulence. And so what is L over eta? L over eta from here from here, L over eta will be VL divided by V eta to the cube. correct?
So why do you assume epsilon is constant again?
It's this picture.
Right, so what you put in, you have to get out, basically.
Yeah, I'm just saying energy here is not really dissipated. I don't know why they call it an energy dissipation rate, because it's only dissipated here. Here it's only transferred.
Okay.
Okay, so it's exactly just to
Sorry,
write down.
Why is this the energy?
What do you mean?
Why V R squared
This
to R over R?
is from here. So this is the order of magnitude of this term. The moment you multiply this equation for V, it becomes an equation for V squared half. So you want to make an argument about energies. So if you multiply this by V, you have 3 V's and 1.
Yeah, before you were using the index R as a derivative. I mean, we confuse a bit. So it's not
Yes.
derivatives, right?
No.
So
Okay.
what does it mean? It's the V at that.
It's this picture, so I'll call this VR.
And that's VL, no?
Yeah, if this is L, L is here, right? So
Velocity, right?
yeah, let's be slightly more formal, and we'll say that. So I'm going to make arguments only about isotropic homogeneous turbulence. So no walls. Everything is, all of the boundary conditions are periodic, okay? And VR, in fact,
Care about the dimension we're in?
yes. This also applies in 2D, but it goes in the other way.
Right.
So
we're
it
in
changes.
3D.
We are in 3D, so let's write it down. 3D homogeneous isotropic turbulence. Okay, and I wanted to be slightly sloppy until we got to that point, but the question is right. I've mixed notations, so what the hell is that R? I'm talking about velocity differences at a certain scale R. Okay, that's what that picture is about. What does it mean that I'm at the different scales? It's because I'm caring how different things look at different scales. Okay, so in fact, you can define this formally, and we'll define it here. VR equal, okay, you take the average, and we'll talk about what this average is, of the absolute value of the velocity field at a point x plus r minus v at a point x. Okay? So these are the typical fluctuations at the scale r. The reason why isotropic is important is because I don't care about the direction of this r. V only will depend on the absolute value of r. Okay? So I take velocities in 2 different points. I make their difference. I'm doing the absolute value. In fact, the formal way to do it is scalar product. So this should be the, but let's forget about that. It doesn't matter for the arguments I'm gonna make. Okay?
Sorry, so your condition 1 is it's actually Reynolds number equals to 1.
No.
Why not?
No, it's the Reynolds number at V eta, which is equal at eta, which is equal 1, which is right. So you're saying
But I mean,
at
the
eta.
Reynolds number always depends on the scale at which you're looking at.
Yes, of course.
So that's
So
the same
so
condition.
Reynolds number in a system where things depend on scale, but in general you need
No,
to
I'm saying
fix
yeah.
a length scale and a velocity scale that are relevant. And what I'm saying is that if the Reynolds number
It's the
based
same-
on the large
it's
scale
the-
is large.
It's the same argument you make to calculate the Reynolds number in the first place. It's
Yes.
the same equality, you just replace u by v
Of course.
and lambda by eta.
Of course, yes. So you could talk about Reynolds number as a function of length scale. Here, this number is based on the large scales, and that is the relevant number to talk about this.
And when I'm in a plane, where does the energy injection come from?
Well, the energy in the atmosphere comes from pressure gradients, typically, or temperature gradients that impose.
So here we don't care about
Grid.
that, we just say there's an energy.
Yeah, we care in the sense that there need to be energy injection, otherwise this will die.
That comes from that term, the delta.
This
delta,
F.
The F.
Well, you have to input, like this is just a Lagrange multiplier, right? So if you don't force P to be something, so you can either put in boundary conditions for P, or you can put a forcing, and they'll both do the same thing. Okay, so just let's finish just this argument, and we'll keep going tomorrow. So U divided by V, so we have eta, and from this relation we can get V and tau eta. So let me write this down. So we have eta, which is epsilon, that's the minus, right? Minus 1/4 nu to the 3/4, and then we can get V eta from what's the best argument? V eta, the best argument is to take this equation, which let's call it star. So you get V eta from star. And that is epsilon times epsilon to the 1/3 times eta to the 1/3. Okay, and so if you put eta in, now we know what eta is. That gives you the relationship for V eta, which is nu to the 1/4, yes, and epsilon to 1/3 minus 1/12, which is 4 minus 3, 1/4. Correct? Yes. Great. Okay, so now we can put it in here. Okay. And what does this give me? So let's say this is U to the 3rd, nu to the 3/4, epsilon to the 3/4. Okay, I'm rewriting things. Let's write it down as U divided UL divided by nu to the 3/4. Okay, because I know this is the answer, and then everything that comes out should be a 1. I have a U to the 3rd minus 3/4, which is 12 minus 3, 9. And then I have a nu to the 3/4 was okay. Epsilon to the 3/4, I need to put it here. And then I have an L to the 3/4. Okay, and I hope this is right. Correct. Okay, this, by using the same relationship that's there, it's 1, because it's the velocity to the 3rd power epsilon L. Okay, there is a 1/4, but forget about the 1/4. Okay, makes sense? Okay, what is this? Reynolds. Okay. So I think this is a good point to stop, because we just demonstrated that this picture occurs at large Reynolds number because injection and dissipation of energy are separated. So now we understand why elephants are different from E. coli. Things occur, energy is injected at a much larger scale for a turbulent field, and a lot of things happen in between. Okay, E. coli lives here, so he doesn't know about it. Okay, so I think this is a good time to stop.
And for 2D, it's 2/3.
For 2D, it's not 2/3. I think it might be the same, but I should check.
I just have have a vapor collection.
It could be 2/3.
Two-thirds turbulent and 2/3 Kolmogorov gas.
No, minus 5/3.
Okay.
We'll talk about 5/3. That's the spectrum. Okay, thank you.